All right, John, you think we should get started?
Sure, yeah, let's go for it. I guess if anyone else pops in, you can admit
I'll
them
take
as
care
we
of
go.
everything.
Yeah. Well, thank you again, everyone, for coming today. I'm John Harkness. I'm the CEO of Rewire Neuro, and I know a lot of you from the help that you've given us over the years and the time you've spent using Pipsqueak. So first off, thank you all so much for being here, and thank you for being supporters of what we're doing. The letters of support that a lot of you have written for our grants are extremely helpful, and just the time that you've put in to help us make this software platform the best it can be is extremely wonderful. So thank you so much. So today, I really just want to run through some of the improvements that we've been making over the last few months. A lot has changed in Pipsqueak since it was first released in 2016. And so today can be a little bit of a guidance on where we're going and what's different now to help you get going with it. We're going to set aside about 20 minutes to just talking about some of the accuracy results, what Pipsqueak can do. And then we're going to switch over and look at some of the ways to do that analysis. We'll actually go through a couple demos. And so during that time, if any questions pop up, feel free to just interject and ask me what's going on, because there's certainly a learning curve to getting some of this going. So with that, let's start. So a little bit of context. Rewire Neuroscience is a company that I started while I was at Washington State University doing my postdoctoral fellowship there. And Rewire is really focused on developing the Pipsqueak software that came out of Dr. Barb Sorg's lab at WSU. So if any of you have seen our first publication about Pipsqueak, I think it was in 2016, we really just had this initial concept of what Pipsqueak could be. It was helpful for us. And since then, it's grown quite a bit. And so I've actually directed a lot of my time to developing Pipsqueak and getting Rewire going. And so now we're happy to devote all of our focus to really making this the best product that it can be. So the team that we have doing this is really amazing. I think one of the best things about Rewire is that we've really organized ourselves to be much like an academic research lab. Everybody here is well-versed in the science behind what's going on, and we're really focused on ensuring the scientific credibility or capability of Pipsqueak software. And so I'm the CEO, and I've also got on the call here Christy Lawton, Dr. Christy Lawton, who is our Chief Science Officer. And Christy, I'll let you introduce yourself.
Oh, thanks, John. Yeah, as John said, I'm Christy Lawton. I'm the CSO of Rewire. And as a grad student, Pipsqueak, for me, Pipsqueak didn't exist, and I definitely would have liked to have had it back then. My background's in neurobiology and genetics, and I did quite a bit of my own share of rather painful manual image analysis. So I'm really excited to be involved with this company and involved in helping develop this tool that'll hopefully make all of our lives a lot easier.
And so in addition to us, we've got a really fantastic group of software and machine learning developers. So whereas I did a lot of the early programming for the Pipsqueak macro, there are now much better programmers than myself who are really making this thing shine. And so I'm excited to show you some of what they do. We also have a great group of advisors, and I really want to point out Barb Sorg, because I think a lot of you are probably familiar with her research. Barb has really been just a guiding light for us in terms of how to develop Pipsqueak and where to take it. And now it's neat to show her where we're taking it past my postdoc. So with that, let's take a look at really what Pipsqueak is. I think the story really began back when I was a postdoc. I joined Barb's lab, and I was, oops, yeah, there we go. I was this really young, excited postdoc. I got to join her lab and do this interesting research on perineuronal nets and cocaine. Oops. And this was a really exciting time to me. But shortly after starting in Barb's lab, she asked me to reanalyze a bunch of the graduate students' confocal micrographs. So this was Megan Slaker. And so what that meant was that I would be counting years of Megan's confocal images all by hand. And as I think a lot of you know, this task that meant that I would have to manually locate and circle thousands of these cells and then co-localize all of those cells between a number of these multiplex biomarkers, all by hand. And so here's my, this is my introduction to being a postdoc. So 2 months later, I finally finished counting all of these cells. And I think in total, there were probably about 10,000 cells that I counted in that analysis. And as I was preparing to triumphantly show Barb how productive I'd been, I noticed that there was a problem. And that was that while Megan and I had both done the exact same analysis, we came up with significantly different results. And so that's really the basis of what we published in 2016. We actually looked at two different methods. We looked at a point method where we measured a number of points around a perineural net. And we used a region of interest method where we circled the perineural net with a bounding box, as I think a lot of you are more familiar with. And so, you know, these differences in both the intensity and the total number of cells that we quantified, I think goes to show something that's been talked about and published about quite a bit recently, and that's that somewhere between 50 and 70% of research can't be reproduced. And some of that is due to variations in the protocols that we all use, and some of that is due to bias and error that creep up into analyses like this. And so all of it reduces our ability to publish papers and secure funding, and ultimately that slows the development of effective medical treatments. And, you know, as we're seeing right now in this pandemic, that affects everybody's lives. So who was right, Megan or I? And the problem with this sort of thing is that there really isn't any way to know which one of us did the correct analysis without doing it for a third time. And so as I was preparing to find a new job that night, I was searching the internet, it occurred to me that I could probably actually write an ImageJ macro that would help with some of this analysis and to make it faster. And so that led to the development of Pipsqueak, the original version of Pipsqueak. And what we found and what we showed in the 2016 paper is that it improved both the intra-rater and inter-rater reliability using this region of interest method. So it did two things. It really tested our protocol, and it showed that a number of people were able to produce the exact same quantification over time. So that was really fantastic. This is also where Pipsqueak got its name. So it is initially an acronym that stands for Perineural Net Intensity Program for the Standardization and Quantification of ECM Analysis. And I think the K is silent in that acronym, so you'll just have to trust us on it. And so, as a lot of you are probably aware, this macro is really great at detecting and analyzing these perineural nets until it wasn't. And one of the reasons for that was that it would struggle with image format and background staining, or even if two cells were too close together, it would have a hard time pulling them apart. And so 5 years later, I think I'm still working on making this time-saving software, but as you're all likely aware, it's come quite a way since then, and we have now called it Pipsqueak AI. And so Pipsqueak AI really takes the old Pipsqueak concept and puts it into an artificial intelligence platform that automatically detects our biomarkers. Pipsqueak is now a deep learning system that can improve with experience, and I'll talk a little bit about that in a minute. But as we've built it, our AI servers are in the cloud, and that helps us begin to grow beyond what we can do right now in ImageJ and Fiji. And I'll show you more about that in a second as well. So over the last year, we've annotated somewhere around 4.5 million cells, all with the intent of training around a dozen different AI models or a couple dozen AI models to detect around 15 classes of biomarkers. And so here I'm showing you 4 of our most commonly detected cells: WFA, parvalbumin, CFOS, and 8-oxo-DG. And you'll notice the similarity between the human annotations in white and Pipsqueak's annotations in green. And so what we found was that Pipsqueak significantly improves the accuracy of cell detection in each of these classes compared to manual analysis. And what you're seeing here in the bar graphs is actually a number of different AI technologies that we've experimented with over the last year. And so we've now settled on something that significantly improves the inter-rater reliability of analysis of each of these biomarkers. And we can actually go one step further and quantify that that we're able to improve the accuracy between 8 and 26% down here at ADOX between better than manual analysis. And that's when we're letting Pipsqueak run without intervention. And so as I think a lot of you are aware, we tend to use Pipsqueak in kind of the semi-autonomous mode where it gives us a chance to go back and double check ourselves. And so we're still quantifying that. And actually, this is going to be the point of an upcoming publication and our presentation at SFN, virtual SFN this year in January. But right now, we're estimating that when using Pipsqueak AI and doing manual or semi-automatic double check at the end, we're improving accuracy greater than 98%. And so this is really exciting. But even on its own, here we're able to produce about 94% accuracy in WFA, 94% in PARV, 98% in CFOS, and 95% in ADOX. So really exciting results, especially seeing where Pipsqueak was just about a year ago. So in addition to inter-rater reliability, we really wanted to test whether Pipsqueak was able to improve the intra-rater reliability of an image analysis. And to do this experiment was actually a little bit more complicated than it might seem at first glance, and that's because Pipsqueak is going to return the exact same cell detections each time we show it an image. And so intra-rater reliability would appear to be 100%, which doesn't really make a whole lot of sense. And so we decided to test this by showing Pipsqueak and our human raters the exact same image rotated 4 times, and then to compare scores between those rotations. And when we did that, we found that our raters were actually fairly inconsistent at counting and quantifying these red blood cells, but Pipsqueak was really spot on each time. You can see that over here on the left side of the graph. And so to put a finer point on that, what I'm showing you here is actually the intensity score of these red blood cells that were analyzed. And I think that's important because with the intensity score, it's taking into consideration the bounding box that's placed around each cell. And so what's impressive is that each time we rotate this image, the neural net is actually seeing a completely different picture. It has no concept that it's the same image, and yet it's able to produce almost identical results. So I think that really speaks highly of what our AI is capable of doing right now. And then, of course, I think much to the relief of graduate students and postdocs everywhere, we're able to do this quantification in about 1/1000th of the time it takes to do by hand. And so whereas it took our scores around 7 hours to analyze these 20 images, Pipsqueak was able to do it in around 4 minutes. So, just really no competition between the two there. And so lastly, before we get into some demos, I just want to highlight some of the Pipsqueak AI system and point out where it's the same and where it's different from Pipsqueak without AI. And so we're calling Pipsqueak without AI Pipsqueak Basic. And this is really the exact same software that you've probably had some exposure to over the last couple of years. Only now we have a professional team of developers who are supporting it and improving it. And it really is becoming, I think, a very solid piece of software. So Pipsqueak, at its essence, uses thresholding and object area size to detect and filter cells. And so when we're going through our double, single, or triple label analysis, we're using that thresholding to detect cells. And I think it's a fantastic method for some applications, but it really struggles in those high background settings. And so one recent improvement that you'll see with Pipsqueak, or actually all improvements that will show up in Pipsqueak AI will be present in Pipsqueak Basic, including some interface improvements, except for the fact that the Pipsqueak AI really brings in our artificial intelligence to the analysis protocol. And so as I've just shown you, the AI is improving both our speed, accuracy, and precision of analysis. And it's actually able to bring in some features that aren't capable without AI, such as quantifying large tiled images in a single pass, and I'll show you that in a second. We're also releasing frequent updates to the accuracy of our model, and we're continually trying to release new models that will help you detect new biomarkers or different biomarkers. And then coming soon to that point, we're actually releasing the ability for you to train or tune your own AI models that will be specific to your images. And so I think this is going to be a really fantastic way for the analysis that you're doing in your lab to, really be as easy as possible or as automatic as possible. And so to put the difference between these two into a little bit more of a perspective, we think that on average, users tend to analyze between 60 and even 560 images per month. And each of these images, takes around 15 minutes to do, I think, in a trained person's hands. And so that equates to something like between 180 and over 2,000, like 2,300 hours per year of analysis. And what's important to recognize there is that a person working 40 hours a week for a year puts in around 2,000 hours per year. So I think when we talk about image analysis being a full-time job, it literally is. And so if we're to figure out a cost for that sort of analysis, it actually costs the lab somewhere between $4,300 and almost $60,000 a year to do image analysis. And that's without considering the time it takes for the PI or the manager to work with those individuals, or the time to redo an analysis if it goes wrong, such as when I was redoing Megan's analysis. And so the AI, is extremely efficient at doing this. And what we're seeing is it's reducing the time of analysis, down to just a fraction of that. So conservatively, if Pipsqueak is reducing image analysis from 15 minutes per image to 2 minutes per image, and I think it can actually run quite a bit faster than that without intervention, we're reducing the time to somewhere between 24 and 200 hours per year. And that is saving a lab ultimately between $3,000 and $50,000 a year, even with a Pipsqueak AI license. And so, I think we're really excited about what this can do to improve both the speed and accuracy, but also, I think just in terms of a lab sustainability point, this is a really fantastic tool. And so with that, let's switch over and take a look at some of what Pipsqueak can do. And I'm gonna move over to a new screen here. Anyone have any questions at this point before we pop into some demos, or anything they're hoping to see in the demos? So I want to get going with Pipsqueak, and I actually want to start from the very beginning. And what I'm going to do here is actually register as a new user, because I think this is probably one of the first areas that can be confusing when getting going with Pipsqueak. So what I've already done is purchased or I've got a Pipsqueak basic license or a Pipsqueak AI license from the Rewire Neuro website. And so I've received my email here with my order ID and my license number. And so after I've installed Pipsqueak into Fiji or ImageJ, I've opened it from the menu bar, and now I'm going to register as a new user. And so when I do that, I'm going to have a window pop up here that allows me to put an email address in here. And so let's just come up with a new email address, and I'm going to enter my password twice. And what's important to remember here is this is actually a different user account right now than what you may have used to purchase your license on the Rewire website. This is something that we're just in the process of developing, and so at some point you'll have a single sign-on that will carry you all the way through this. But for now, you're welcome to use the same credentials. You just have to recreate a new account here as you go. And so great, let's put in the new license that I just got here. So you can see I was just playing with this a minute ago, but all right, we've got the order number here in the top field. I put my license number down here in the bottom field, and I'm going to register a new account. And in just a second, we should see that it pops up. Great, registration was successful. And so now when I come back, I'm just going to log in with the same password. And when I say OK, what we should see here is that our main menu pops up, and this is really going to be our home base for a lot of the analysis that we're doing. And I can see the version of Pipsqueak I'm using. It was actually just released last night. We had a little improvement that we put in there. I can see and I can see that my AI license is active. And so I'm all ready to go. So what I want to walk through is actually three different types of analyses that I think a lot of people are running into. And we don't have a whole lot of time to really dive into a lot of the settings or even specifics of this analysis. But what we're planning to do in the future is come back and go through some more of these specific steps in greater detail. And so if that's interesting to you, watch for these webinar announcements and join in. Additionally, we'll be posting more of these videos on our Rewire website, so you should be able to see some of the steps as you're going through them and just watch those videos. So let's start off here doing a single label analysis. Just briefly, I'm going to take a look at the settings I have in here because it's important to make sure that I've really got it set up for the type of image that I want to analyze. We're going to look at an 8-oxo DG image, and really a lot of what's here in the middle is now very specific to the analysis that you'll be doing in a Pipsqueak basic license. So when you have the AI enabled, we actually don't have to worry about setting the threshold levels to make sure that a cell is being detected because our AI is doing it here automatically. Some of these menus are going to improve over time and we'll see some stuff streamlined, and I'm actually excited to show you where we've already done that. So for 8-oxo DG, what I do want to focus on here is that I like to analyze, or I like my ROIs to be circular. And I'm actually going to set my ROIs to just be exactly as they come in from the AI. I'm not going to enlarge or shrink them as we've talked about in the past with some of our WFA staining. So great, I've got that. I'm going to take a look at one more setting here. I'm going to look at my AI settings, and this is because I really want to make sure that I've got the appropriate background subtraction set up for the type of image that I'm analyzing, and also taking a look at what we can do with the AI. So quickly here, there are some great detailed steps for rolling ball radius over on the Pipsqueak.ai website, and I'll show you that here now, just where to find this. If you go to Pipsqueak.ai, you can go to the Learn More tab, and Frequently Asked Questions now has some really fantastic instructions on some of these more specific aspects of Pipsqueak. And so I'd invite you to go check those out. Briefly here, what I'm going to set for my rolling ball radius is essentially a pixel diameter of the largest cell that I expect in my image. And so we're going to see that in two different ways here today. The first I'm just going to put in is about 30 pixels, which seems to be accurate for, or appropriate for the part, or the 8-oxo image that we're going to look at here. I'm also going to set my background subtraction at low. And if you'd like to know more about high versus low, again, I'd invite you to look at the frequently asked questions sections. But generally, what high background subtraction is doing is keeping the image background slightly brighter than it would be with the low setting. So with the high setting, we're actually subtracting the brightest third of the ROIs, or background ROIs that we collect in our image analysis, and the dimmest third. Whereas with the low setting, we're actually subtracting the brightest 3/4 of the ROIs that we're using for background subtraction. I'll show you what that means in a second, but again, I'd invite you to read the frequently asked questions section. And so lastly, with our AI, we now have the ability to choose exactly how it's running. I'm going to tell you a little bit about speed versus accuracy, but you can see how I've set it up here initially, and image subsectioning is off, and I'll tell you why in a second. So great, let's do our single label analysis. As I come in here, I have two options. I can analyze images, or I can sum Z-stacks of images. And we're not gonna have time to go into Z-stack summing today, but again, this is something that we can go into in greater detail in the future. And so now I'm gonna choose my first image. The menus have changed here a little bit. I think this is a great improvement now that we're actually showing you where you're opening your image rather than kind of sending you off to find it on your own. This menu is going to improve a little bit more as time goes on, and I think you'll see that what we're doing is really making this as intuitive as possible, so that you can start to name your images anything that you'd like, and ultimately bring them together for co-localization analysis. So I've got my image in there. I'm going to say OK, and it opens up. I've now got some new settings, or some new options to add in more detailed information about the type of image that I'm analyzing. And the reason that I like to do this for my own analysis is that when I get to Excel and I start sorting my data, it's very difficult to know exactly, which image is from which subject, or which experiment, or which treatment. And so you're now able to enter that in so that it will follow you all the way through your data quantification. As you'll see going forward, we're going to start making this data actually available to you online. And one of the reasons that we're doing that is because we can provide some greater data sorting capabilities, actually just through database sorting of the data that you're producing here. And that's going to make it a lot easier when you've got 50,000 cells in an experiment to really just find the group subset that you're looking for. So in this case, we'll just say that this is experiment 1, this is subject 1, and this is treatment 1. This is my very first experiment, or first rat here. And so as it opens up, I have my background subtraction options. So we talked about the level of background that's being subtracted, but one of the new things here that I want to point out is both the ability to limit the area of our analysis. And so this would allow me to subsection down maybe a white matter track that's going through my image, or the side of the brain that I don't want to analyze. In this case, I'm going to take the whole thing, but I'm going to show you that we can manually edit the 22 boxes that are being placed around the image. And I think this is a great improvement. In this 8-OXO image, it's really hard to actually get the background subtraction boxes off of the ROI, or off of the cells. But I can now nudge them around a little bit. And I think this is a great way to really ensure that the background you're capturing is what you're looking for. And so again, to really focus on what's happening here in terms of the background ROIs that we're using, since I'm using the low background settings, I'm actually going to only use around 5 of these ROIs, to the 5 dimmest ROIs, to subtract out my background. And when I do that, that background subtraction is really only being carried over into my image quantification, whereas our rolling ball radius subtraction is being used to ultimately smooth out the background in this image. So I've got those, I like that, and I'm going to continue forward. And so now I'm already at my AI menu here, where it's going to allow me to detect the cells. And so we've got a couple options. We can choose the detection sensitivity. Essentially, as I move this lower, our AI is going to be more conservative in the ROIs that it returns to you. I can also choose how much of an overlap would be too much in my preference for the ROIs returned. And so in an image like this, that's actually a pretty big consideration, because a lot of these 8-OXO DG stained cells overlap quite a bit. And so I'm gonna lower my threshold a little bit, because I don't want too much overlap. I can choose the model that I'm using here, and in this case, we've got 4 different models that are ready to go out of the box, and we've got this other model that's a really great general cell detection AI model. And so if you're coming in with something that maybe looks like CFOS, for example, DAPI, but DAPI isn't available here, I would suggest you use other. And as you'll see in the next couple of weeks here, we're going to be starting to release a number more models for you to choose from. So great, I've got all that set up, and I'm going to say okay. And so what we're going to watch here is that the image is now being processed by the AI. We can see a little bit of feedback on what's happening with that. And in just a second here, we see that it's returned somewhere in the order of 350 ROIs, just like that. And so I think this is the part of the AI that's just really, really fantastic to see. I'm going to zoom in on the unsubtracted image here just a little bit so we can see what's going on. And for these cases, for this example, I think I'm pretty satisfied with the cells that it's detected. If I were to go in and want to add a couple more or touch up the annotations a little bit, I could at this point. And so again, this is something that we will touch more on in future webinars. But one of the things that you'll notice now, this is a difference from previous versions of Pipsqueak, is that you're able to modify these ROIs straight from the window right here. You actually don't even need to click into this setting here. So great, we've got a good set of ROIs for this image. And so I'm going to say complete analysis. And when I've done that, essentially the ROIs are being quantified, and we can see our output printed over here in the log menu right to the side. What's great about Pipsqueak is it's also saving those results into the folder where we did the analysis here. And so the image that we just analyzed is this guy here, and And what I can find when I go into its newly created folder is both the ROIs that were produced by analysis. So if I want to go back and audit the analysis that was done, I can do that here simply by dragging the image and that ROI file into the Fiji menu bar. And I can also see my results. And so the results are now saved as a comma separated value. And so this is a great way to just import it straight into Excel. And when I do that, you can see it already split it up into a pretty logical representation of the data. And I can now go through and sort out exactly what I'm looking for, maybe with a bunch of different subjects and a bunch of different treatments. And so, really how to get into that quantification is again going to be the focus of a of another webinar coming up, but I think that's a great place to start with it. So we've got that, and if there aren't any questions on single label analysis, I'm gonna skip forward and actually take a look at double label analysis. And one of the reasons that I think it's good for us to do this is because we have a number of double label images available to try out on the Pipsqueak.ai website. So if you go to Pipsqueak.ai downloads and scroll down to the bottom of that page, over here on the left, you can find our practice image set. This is actually the image set that we use to calibrate our own analysis in Dr. Sorg's lab. We would just have everybody run through this, do these same 20 images, and then we would graph them between the lab. And that'd be a great way for us just to make sure that somebody wasn't thinking that a perineural net looked like something that it wasn't. And so download that there. Let's go ahead and open up one of those here in Pipsqueak. So again, I'm going to go to the Pipsqueak bar or the menu bar and open Pipsqueak. And this time when I do that, it's going to remember my login, which is great. And now I'm just going to go to double label analysis. And so again, I'm going to analyze these images. And now when I do it, I actually have a couple more options. I can choose both of the stains that we're going to look at. And so I know just from having set up these images that the first one I want to look at is parvalbumin, and the image suffix for that image is number 1. The second image that I want to look at is WFA, and the image suffix for that is number 3. And so, this is I think the one part of the program that I'm really excited to improve here over the next couple of weeks, because as I think a lot of you are aware, the naming of your images for double label analysis really matters in Pipsqueak. And so what I mean by suffix is we're going to analyze this image here. And when we do that, Pipsqueak is going to look for the one that has an _1 and an _3 at the end of it. It's also looking for _2, and that's going to be the merged image that we'll look at the end. And so, while this is a really fantastic way in Dr. Sorg's lab for us to do this analysis, it will be a lot easier to just point directly at the image you're looking for. So great, we're just going to open up the very first PARV image here. And so I'm going into that practice double label set. Let's see, I'm looking for my images, and here it is. Let's just do the very first one, _1. And so one of the things that, all right, so we've got our chance to enter some info there. Again, I'm just going to skip it here. But one of the things that Pipsqueak is now allowing us to do is to double check the images that we're opening. And so it's popped up this menu here. It's actually it's printing out the images that it's looking for. So the second one is going to be my WFA image, and there's the _3 that I'm looking for. If this wasn't correct, I can now actually say no and go back and choose the correct ones, or maybe change my suffixes. In this case, it looks great to me, so let's move forward. So again, we can do our background subtraction. I'm going to show you manual selection here real fast. And so in this case, it gives me the option to just freeform choose the areas of my image that I want to use for my background levels. And so I'm going to choose these 3 here. I think that's great. Maybe one in the middle too. So for example, that's a great way to do that. Great, so we've got our PARV image ready to go. It actually remembered that I'm doing parvalbumin here first. Let's take these detection settings and let's move forward. And so it's run analysis. Voila, we've got that PARV image pretty well detected. And again, going back to the double or the semi-autonomous mode that I think is really smart to use in all sorts of analysis, I'm gonna toggle through this here just to double check the cells that Pipsqueak popped up. And one of the ways that I can do that is by taking either the subtracted or the non-subtracted image and pulling up my ROI manager. And I'm just gonna click the show all button. And when I do that, I can see exactly, oops, come back here. I can see exactly where the ROIs are placed and where they're not. And for this image, I think that that actually does a really good job. I see that there are two parv cells here that are bumping up against each other, and it's got both of those. Maybe there's actually a little cluster up here that's too clustered. I think it's put one in the middle of something that's probably just two. So let's delete that, and we can toggle again. And I think that looks pretty good. Let's go for it. So we can say complete. And it's going to quantify that image, and now it's going to actually automatically open up the WFA image. And so let's just take automatic boxes around the side for sake of time here. And so we're already ready to do our WFA image. So let's take these detection settings. Again, I'm just going to leave these accuracy and image subsectioning as they are. I'll talk about them in just a second. And let's analyze it. One of the most impressive things that I think our AI can do is these large, heavily clustered images like that ADOXO I showed you, and then WFA. And one of the reasons that WFA is really impressive to me is that I think, as a lot of you are aware, WFA cells are really hard to put an exact meaning around, or an exact definition around. And that 2016 paper, I think, goes a long way to talking about what we consider to be a full perineural net WFA setting cell and what we don't. And one of the things that we look for is just exactly how much of the cell is being encompassed by the WFA stain. We tend to use about 2/3 as our threshold, but that's obviously not an easy thing to exactly quantify as you're looking at these images. So when we look at the sort of ROIs that Pipsqueak put on here, I'm really satisfied with it. One of the things that I think is great that I want to point out is that it got these two cells here. This is probably a gap junction between these two perineural nets, and it got those two there. Maybe there's some smaller ones in here that would sort of be on the threshold of deciding whether or not it's a net. I'm seeing that guy down there. But this is really where it starts to move into what your lab has defined as being a perineural net. So let's go ahead and take that and move forward. And lastly, what it's going to show you here is going through and looking for the co-localization of the parvalbumin and the perineural nets. And if you've used this before, I really am excited with how much better our co-localization runs now. We improved our algorithm, and it's just incredibly fast, and it's incredibly accurate. And so, just kind of looking at this here, this is again our chance to double check what Pipsqueak has returned. I think I'm really satisfied with all of these co-localizations. Again, this is something that we can go into in more detail in a future webinar, but one of the things that I'm looking for is just to make sure that my parvalbumin cell is smack in the middle of my my perineural net. And so, there's a lot of gray area there sometimes. And I think one of the problems with co-localization analysis is that it's really easy for our eye to trick us into thinking that a co-localization is there. One of the ways that I like to think of this is that if that cell wasn't detected in single label analysis, that single channel, then we really don't want to include it here in double label analysis. And so, you can definitely spend time going back and auditing what's here. But for example, these two cells were not detected as being co-localized. And I think we really have to trust that if we didn't detect that, maybe parvalbumin in this case, in single label analysis, then we don't want to include it in double label analysis. And so with that, let's go ahead and accept those. And again, it's printed out all of my results here over in the log. And one of the things that's great about double label analysis is I've now got 4 data sets from these 2 images. I've got my single label WFA, my single label parv, my single label WFA, and then the double label quantification from that subset of cells that was co-localized. And so if you don't have any questions with that, I want to show you one more thing very quickly, because I think this is really exciting. And that is our ability to do a large tiled image with our AI. And so I'm going to go back into single label analysis here. And I'm now going to open up an image that was actually sent to us by one of our users. And so maybe you'll recognize your image if you're on the call. And what I've got here is actually a tiled image of, I think, what is parvalbumin. And so we're going to open that up here. And as you can see, this is an enormous image. This is like 5,000 pixels wide. I'm going to skip this here. And I'm actually going to, I'm going to make that image a little bit smaller so that we can see it because it's so big. There we go. And so, you know, one of the problems with an image like this is it's really just beyond the capability of Fiji to go in and find those cells through thresholding. And it's actually even really hard for, for our AI to do a big image like that. And so we developed a feature that's actually able to reduce the size of the image as we upload it and chop the image into subsections. And so I'm going to do that here in my AI menu by going to change settings. And now I have the ability to see all my menu settings. I think this is a great improvement. I'm going to go to AI settings. And here I'm going to change from accuracy to speed. And I want to be really clear. The reason that we call this accuracy versus speed is because we're changing this TIFF image into a JPEG. And technically, when you do that, you're reducing image information. However, this image is only being sent to the AI server to produce our ROIs. And so this is not used in quantification. So when we reduce it to a JPEG, it makes it faster for it to upload. It has the potential to reduce or change the quantifications that are returned, but we've actually never seen a difference experimentally. I just want everyone to be aware of what's happening. And so this is why we call it speed versus accuracy. So in this case, we're going to go speed. And first, let's actually do this with subsectioning off, and I'll show you what happens. So, all right, great. We're going to analyze this parv image. And when we do that, it's uploading. And it returned one cell. I don't think that's right. So I don't really like that. And so let's actually go back, and we're gonna rerun this detection, and I'm going to change the setting. I'm going to change my AI to be subsectioned. And here, this is a really big image. And so I'm going to use our 6x6 feature. This is the most aggressive subsection. And essentially what it's going to do is it's going to divide the image into 6 horizontally and vertically and send it back up to the server. And so when we do that, what we should see is a much better detection. So wow, that's just night and day. I think this is one of the most exciting things that we've developed in terms of what our AI can do. And so what you'll see here is, you know, it actually was incredibly accurate. Oops, that's kind of dark, but incredibly accurate at detecting a lot of those small cells here. This is just much smaller than what our AI was trained on. You know, there are a few out here that are probably outside of the tissue, maybe some specks on the image that I would want to analyze or fix by hand. But, you know, I think this is a great example of what can be done and should make you hopeful about what you can potentially do with some of your large tiled images. So great, let's accept that. And we can finish that image. And so that's actually the end of what I want to show you today in terms of demoing new features. And so let's use the remaining 15 minutes here just to answer any questions that you have or any specifics about the type of analyses that you're doing or things that you're curious about. And if there aren't any questions, then I am happy to run through some additional things that
John,
we have set up in Pipsqueak.
We did have one question in the chat.
Can you see the chat? I'm actually not able to see that there. So I see, is it still necessary to put image size and pixel information somewhere into the program? Yeah, that's a really great question. And so we do still have that ability here. And actually what's being changed when we change the image dimension in our settings is the quantification of area that's being put into our results. And so this actually isn't going to change the way that the cells are detected. Our AI now doesn't care about the image diameters of your picture in the same way that thresholding does. But it will change the area quantification that's laid out in your results. And so if you want that to be accurate, then you definitely need to go in and make sure that you're changing this number. But you know, as we saw, I didn't change that when I looked at the tiled image compared to the very small, 300 micron image that we collected in Barb's lab. The other things that are in here, of course, are the unit of length. I think for most applications, you'll probably always be looking at microns, and so you can leave that there. But this is a great time to talk just a little bit about the the Z stack image summation. So one of the things that we did in Barb's lab was to collect our images on a confocal microscope. One, because it allowed us to detect or to image a number of different channels and co-localize or multiplex these biomarkers. But it also allowed us to collect a deeper set or a deeper stack of tissue so that we might be able to encompass an entire paraneuroma net. When we quantified, we would always collapse our images into a two-dimensional plane. One, because it was easier for us to quantify it, but two, actually, because we didn't have a way to quantify a 3D image. And so our Z summing step was how we did that. And previously, when you summed one of your Z-stepped images, you would go in and select the number of stacks in there. Now we actually don't need to do that anymore. And so I'll just briefly show you here how we can, oops, I'm gonna go back and do a triple label analysis, but I'll briefly show you how we can sum these images, because this is really one of the most time consuming steps of analysis, I think, if you're collecting Z stacks on a confocal. So I've exported the images as TIFFs from my confocal, and I've brought them into a file browser in a way that allows me to put a bunch of different subject images into one experiment folder. And so in this case, you can see my subject images contains 3 different channels, yeah, it includes 3 different channels, an emerged channel, and it includes, in this case, 20 Z steps. So I actually don't have to tell Pipsqueak anymore how many channels there are or how many steps there are. All I'm going to do is I'm going to select that experiment folder. So I'm going to this parent folder. And when I do that, it opens it up, and you'll see it's just extremely fast going through. So I can double check it. And here I'm going to choose batch mode off. If you choose batch mode on, it essentially doesn't open the images, so it doesn't flash in front of you, and it's a little bit faster, but I always think it's kind of fun to watch them go through. And so voila.
John, just a note on time, we have several more questions.
Images. And so now when I open up my folder again, I can see, here are those images that were just summed. And so that's a, really fantastic way to do that. So let's see, I see another question here in terms of how do we subsection an image for counting. And so the way that we can do that here is, let's open up a single label image. Actually, this is a feature that's only available in SingleLabel Analysis right now. And so let's go back to one of our, I think this is going to be a PARV image here. Or no, this is a WFA image. And so great, I've got this. And now let's say I have a big white matter track, or I just know a really specific region that I want to analyze here. And so to do that now, in my background subtraction menu, I'm going to just click this little toggle button there, and it gives me a chance now to subsection my image. And so in this case, I'm just going to draw right on the image and subsection it, I don't know, something like that. Once I've drawn, the one important step here, this is an ImageJ specific feature, is that we're going to have to add that ROI to our ROI manager. And so I can do that either by pressing the T button on my keyboard or by pressing it, the add button here on the ROI manager. So great, I've done that. I can double check it by toggling it. And I'm going to say okay. And so now what Pipsqueak will do is carry this subsection image all the way through, and we'll actually have a record of this image saved in our output folder. And so this is a great way to now double check, exactly what was done, or even to compare between images. And so, here we can see that run real fast. Let's change it to WFA. Oops, we don't want to subsection this though, because this is a small image. And you can see it run pretty well. So great. Let's see, Christy, actually go ahead and chime in here if I'm overlooking questions, because I see a couple of these coming in.
Yeah, there's several. I think Michelle's question was next.
Itself using Pipsqueak.
So Michelle asked, is there a way to compare the image to an atlas?
Yeah, let me know, whoever asked that question about subsectioning if I didn't fully answer it. And again, this is something that we can go into more detail in a future webinar. But generally, you can see it was able to limit analysis or limit detection to just that region. And so that's fantastic. This is something that will be available in multiplexed analysis going forward. We just initially released it in SingleLabel Analysis. Okay, yeah, so I see a question here about opening the image in Pipsqueak. The image is always black, only when I open it from a 12-bit picture. Would that be okay? As I quantify pictures with 8-bit, I'm wondering if there's a way to fix that. And so, yeah, one of the things that Pipsqueak used to require is for images to come in a 32-bit TIFF format, and that's something that's actually no longer required. So now Pipsqueak is able to open the native format that your image is coming in, which is still probably TIFF, but it will analyze a PNG or a JPEG, and it's able to look at anything from an 8-bit to a 32-bit image. The reason for that is when image J goes through its thresholding step, it's really restrictive on the type of bit depth that an image has. And so we're artificially creating our images to be 16 or 32-bit depth at that point. It actually isn't adding more bit depth to the image, but it allows our thresholding to run. And so in this case, Pipsqueak now gets around that. This is a great addition or a great feature of the AI. And so you can open them exactly as is. If they're coming in as black, I think one of the things, that's probably the first place to start is taking a look at how the images are being captured, either on the fluorescence or on the confocal microscope, and making sure that you've set your laser or light source power up to a level that's giving you sufficient signal. If the image is coming in without a whole lot of signal to begin with, then there isn't a whole lot that we can do with it once it gets into ImageJ or into Pipsqueak. But if that's a specific question that I haven't answered very well, feel free to reach out to me by email or chime in here now and help point me in the right direction, but we can definitely go into it in more depth. So yeah, Christy, I can't see the chat here, so feel free to tell me what I'm missing. Oh, there it is. All right. And so I think, oh, go ahead.
Go ahead.
You've got something, but one additional point of detail to put into our, so we were talking about subsectioning our quantification or cell counting to a specific area. And so one of the ways that Pipsqueak outputs are, the best way that Pipsqueak outputs our cell count is actually in our results. When we're looking at what's printed here, we can actually see the number of cells counted. So in this case, it counted 11. In this case, it counted 114. And so if you're really most interested in quantifying or counting the number of cells in the image, I would just take a look at the highest number image that pops out in the results. If this is a specific thing that you're interested in doing, we definitely have the capability of spitting out that number on its own, just 114 or 11. And to be honest, this is a feature that Pipsqueak used to have. We just didn't find that it was utilized very often. And so let me know if that's something that you would like to see come back. All right, so we're in the last few minutes here. If there are any other burning questions, feel free to pop in. But otherwise, I'd really like to thank you all for coming in and checking this out today. Again, the support that you all have given us over the last couple of years in development of Pipsqueak has just been fantastic. This really is a community effort, and I think we're all really proud to see where this has gone and what it's capable of doing today. On top of that, I think one of the, oops, where's my cursor? One of the most exciting developments we have laid out in front of us is the ability, so I've talked about a couple of these here, but is actually the ability to start taking our analysis online. And when we do that, we're going to improve the interface to a great extent, but it's also going to improve the image analysis speed, It's going to allow us to share images or share files between collaborators and to store those files in a, a deep central repository. And it also is going to enable us to provide better data output. And so, this is something that you'll start seeing in 2021, and we're really excited to share that with you. In addition to fully automated batch processing, 3D co-localization that I've talked about, and this is what I want to get to, is that we are beginning to develop a system that will allow users to tune models to be appropriate for the type of image analysis they're doing. I'd love to hear more input from anyone else who's excited about that feature or has an idea of how that can benefit them.
Along those lines, we have a survey live right now if you guys want to potentially help contribute in that way. It was sent out in the newsletter that we sent out in the last week or two, but I will include the link in a follow-up email to all of you. Thank you so much for coming, and do let us know if you have any other feedback beyond what's covered in the survey.
Yeah, thank you all so much. This is really fantastic to get to see you all, and I'll look forward to talking to you here shortly in the future. All right. Thanks. Bye-bye.